So yeah, many thanks first of all for the invitation to speak at this session. I suppose I'm a slight outlier here in that the systems and models that I'm working on and the diseases I'm interested in are predominantly neurodegenerative, and I'm mainly interested in strategies that mediate neuroprotection. But I think there's obviously a huge amount of overlap with the kind of neuropsychopharmacology field. So I think I'll start in that sense. I'll start a little bit with an introduction to SITraN itself, because you may not all be familiar. We've been open since about 2010. We're now quite a significant research institute, quite a large number of both academic staff and research students and postgraduate students really all working on a kind of major areas of interest: motor neuron disease, Parkinson's disease, Alzheimer's, some significant work in stroke, and also in inflammatory CNS conditions, particularly multiple sclerosis. And last year it was quite nice. I'm contractually obliged to say that we won a Queen's Anniversary Prize at the end of last year for the work at SITraN in terms of improving kind of patient outcomes and promoting development of new therapeutic approaches and strategies to treat these devastating diseases. So that's just a quick plug for SITraN, for those of you who aren't aware. This is what I'm going to briefly talk about. So I'll give, again, a brief overview of motor neuron disease, just a couple of slides, just to orientate ourselves. And then I'm going to focus quite a few slides on how we increase confidence in preclinical to clinical translation, particularly for motor neuron disease. And this is an area that I'm really particularly interested in and spend a lot of my time thinking about and working on. And hopefully you'll see we think we've put in some reasonable systems in place to try and improve that key step. I'll then show in one slide some of the small molecule projects in particular. We do a lot of gene therapy as well. But I'll show you some of the small molecule projects we have, what stage they're at, and then I'll select just two of those, which give some example of how we collaborate. So we collaborate in a whole range of ways, but the two that I'll talk about are a very straightforward project where we collaborated with Sose Heptares with a PhD student, and we've generated some nice preclinical data in our ALS models. And then we'll go all the way to the other end of the scale, a very large program where we've done kind of early target-based traditional drug discovery funded by a partnership with Parkinson's UK. Okay, so just a couple of slides on motor neuron disease. For those of you who aren't aware, it's an adult onset neurodegenerative disease. We have both upper and lower motor neuron degeneration leading to loss of muscle mass, voluntary muscle. Life expectancy is very short, around 2.5 years. Following diagnosis, with death usually due to respiratory insufficiency. So a really terrible condition with a kind of devastating prognosis. I think uniquely for a neurodegenerative condition, there are compounds that are approved that do seem to influence disease progression or to some extent. So riluzol's been approved for a long time, is now generic, but it gives a marginal improvement in survival of about 3 months. It doesn't seem to affect any measures of kind of disease progression, respiratory function, muscle function, and so on. That's approved in both the US and Europe. Edaravone was more recently approved in the US, been approved for a while in Japan. It gives a 2-point reduction on I think it's a 48-point scale of this ALS functional rating scale over 6 months. So it improves the decline in that measure by 2 points. That's not been approved in Europe. So I think uniquely that, to my mind, creates some opportunity for us to maybe follow up and hopefully develop some more effective therapeutics. And we're beginning to see some evidence of that now, particularly for the kind of monogenic subtypes of specific motor neuron disorders. There are about 5,000 people with the disease at any one time in the UK, so low prevalence, but that's because of the very rapid progression and poor prognosis, low lifetime risk, and then more recently, over the past 5-10 years or so, there's been this realization that there's very significant overlap between motor neuron disease and frontotemporal dementia. In terms of disease mechanisms, like all neurodegenerative diseases, it's complex. Many mechanisms have been implicated, and sometimes it's a bit difficult to see the wood for the trees here, but we see a lot of overlap with other neurodegenerative conditions, and some of the pathways that we're particularly interested in are things like neuroinflammation, oxidative stress, mitochondrial dysfunction, and dysregulated proteostasis. But I would point you to this review from Ola Hardeman and some other key opinion leaders in the field. It's quite a nice primer on ALS that covers the clinical features and the kind of underlying pathogenesis as well. So I'll come back to this later because I think this is important, this idea that there are multiple mechanisms possibly active at different times or in different sets of patients. So just to move on to how we at SITraN have gone about trying to improve preclinical screening, and it's really about how do we nominate the right clinical candidates. And for me, the real critical step is then once we think we've generated enough confidence in a particular preclinical candidate, how do we actually take that into the clinical arena, have confidence that we're going to test the hypothesis in the clinic? And I think for me, this is the key aspect really of drug discovery in all conditions, particularly in neurodegeneration. And the kind of overwhelming opinion, I think, in the field would still be that one of the biggest issues we've had is around this SOD1 G93A transgenic mouse model of ALS. So mutations in superoxide dismutase 1 cause motor neuron disease in a small subset of patients, maybe 2 to 3% of patients. But we've known that since about 1993, and we've had models since 94. And much of the preclinical work has been done in this model. And much of the failure of translation, I think, has been blamed on this model. I personally think unfairly. I think some of the blame lies in the kind of over-reliance on this particular model, but I've highlighted 3 issues here, really. So one of them is poor study design. So just, and this has been highlighted in many papers, poor study design has led to an inappropriate control of factors that can lead to bias in results, has led to false positive studies that haven't been replicated. Virtually no study with a small molecule drug in a SOD1 G93A transgenic mouse model where they've looked at survival, this kind of key readout that's been used for a long time, which we're trying to shift away from, has been replicated. The second issue is that the translation from mouse to man is poor. So there's quite a nice paper from Mitsumoto highlighted there. It's a few years old now, but they go through multiple clinical trials and show that even if in very few cases have we measured target engagement in the clinical studies following on from the preclinical studies, and when we have, we've actually failed to demonstrate target engagement. I think that is getting better now, particularly with the recent Tofersen trial in ALS, which I'll talk about right at the end. The other issue, and this maybe is cause for concern, is that SOD1 G93A mutations, as I've said, only represent a small proportion of the clinical population. And the major protein that we find in inclusions in neurons and in glial cells is TDP43, and patients with SOD1 mutations do not have a TDP43 proteinopathy. So there may not be a mechanistic overlap between this and wider ALS. So I'm going to go through those 3 issues and just talk about how we've tried to address them. So the first one, study design. So we, from day one, had a very robust, reproducible in vivo study design, just following really the standard approaches that you would do. My first postdoc was in industry doing preclinical pharmacology, and I think it was there that I really learned kind of how to set these preclinical studies up in a way that we can really rely on the results. So we, from day one, had a defined genetic background with a consistent phenotype, and we introduced a whole range of quantitative measures of disease progression. I'll just highlight a couple of things here. So this is the standard readouts, decline in rotor rod function over time, and time to reach end-stage disease, which is often called the survival readouts, which, as I've said, is not at all reproducible if you don't design the study properly. That's very consistent over, these are studies over about three years. So we've got basically the same phenotype. We've also introduced things like muscle volume measurements, which gives us a really nice quantitative, low variability readout, and those are the kind of readouts we're interested in, ones where we can generate high statistical power to detect treatment effects. Another example is gait analysis, quantitative gait analysis. This is an example from a different animal model that we've characterized, which is based on mutations in TDP43, which we think may be more broadly applicable in ALS. And although the changes we see here in the kind of gait disturbance, and in this model you see this kind of waddling gait, the changes are small, they are consistent and reproducible over time. In terms of how we address the issue of translation to man, again, I think one issue in the preclinical studies, if you look in the literature, particularly from the past, is that maybe the field hasn't done a good job of doing the basic pharmacology. Does the drug get in the brain at the right concentrations to have an effect? So I'll come back to this particular mGlu5 antagonist in some of the later slides, but this is an example where we've done our kind of PK study in vivo in the disease model, and calculated the kind of free drug in brain to make sure that we're going to be able to engage the target. The other really key aspect here is we like the idea of developing readouts that are translatable from the preclinical model to the clinical disease. So we can't do rotarod in a patient, but we can do something like a compound muscle action potential. So this is an electrophysiological measurement which gives an indication of the total functional innervation in muscle. And we see quite profound reduction in CMAP in the QC31K TDP43 mutant mouse model. And here we're showing some data with riluzole where it doesn't really seem to improve CMAP. It does improve repetitive CMAP when you do repetitive stimulation, which is a crude measure of neuromuscular junction health. And that was the only thing that really influenced riluzole in this model. The final thing that we try and do, and again, I'll talk a bit more about this later, is develop novel markers of target engagement. And this is an example, bottom right, where we can measure glutathione levels directly in the CNS and this is a healthy control, but we're starting to do it in patients as well. And that's a really nice target engagement biomarker for compounds that activate this NRF2 pathway, which is something that we're particularly interested in. Just to come to the final point on that slide, which was about validating our models with standards of care. So in the SOD1 model, we've done some work with riluzole. So as I said, this extends survival in patients by about 3 months. But as you can see, this readout that's really not been very useful in terms of telling us whether a compound's going to translate to the clinic, riluzole has absolutely no effect on that, paradoxically, but it actually does slow down the kind of earlier phases of disease progression as measured by, this is running wheel performance. So it gives a very similar kind of readout to rotarod function. And this is at exposures that would be, we think, clinically meaningful. So it's always important to understand what you can measure in the model and how that relates to what you can measure clinically. So it may not be the case that if we don't see an increase in survival with a compound, we won't see it in the clinic, because that's clearly what we see here with riluzole. The third issue that I talked about was clinical relevance. So the SOD1 model may not be, someone raised their hand. So the, I can't get rid of that now, hang on. Okay. Sorry. So the third issue is clinical relevance. So how do we develop, and here what we've done is we've developed new models or validated new models, in particular TDP43 QC31K transgenic model. Am I out of time? No?
5 minutes left, Richard.
5, thank you.
4 minutes, sorry.
Someone kept raising the hand, I thought I was running out of time.
No, okay.
So we've characterized the TDP43 QC31K transgenic, which I've shown you. We've also worked with Jameen Sridharan at King's, and he's got a knock-in mutation in the TDP43 gene in mouse, so a more physiological model. Interestingly, shows a very similar phenotype to what we see with the transgenic, and actually shows some cognitive dysfunction as well, which we think is quite interesting and may relate to this ALS FTD overlap. Another approach we've taken is, and this is work from Laura Frariolo at SITraN, so we've got some really nice patient-derived cell model systems, where we can measure readouts from different genetic forms of motor neuron disease, but also from sporadic disease, rather importantly. So this is the only way really we can truly model sporadic disease. Okay, so just to quickly, in the last 5 minutes, go on to kind of some example projects. So this is the SITraN small molecule pipeline. It's not everything, it's just some of the things that we can maybe talk about. So we have various programs that have either been developed or originated in SITraN, or where we've accessed compounds from collaborators, or programs have been running collaboration. And I'll just briefly go into the SOSE Heptares collaboration here and the Keepstone Therapeutics. So with SOSE, we were really interested in their negative allosteric modulator for metabotropic glutamate receptor 5. So there's a strong evidence for glutamatergic excitotoxicity in ALS, and other people had shown that MPEP and mGlu5 NAM had some efficacy in the SOD1 G93A model, but this compound's got significant liabilities. In particular, this acetylene group can lead to toxicity. It's got poor pharmacokinetics. I showed you the pharmacokinetics for the Heptares compound, which they were able to develop following their really nice STAR methodology, where they actually first to solve this crystal structure, and they could do some really nice structure-based drug design to get away from this problematic acetylene group and get a compound that filled this allosteric pocket really quite nicely. What we were able to do is develop a receptor occupancy assay where we could rapidly evaluate receptor occupancy in tissue from our SOD1 mouse model from efficacy studies. And with doses of 3, 10, and 30, we got a nice range of receptor occupancies, so almost complete occupancy for the full dosing cycle with 30 mg/kg, for example. And that correlated with quite interesting data that we weren't expecting. It correlated inversely with the degree of glial activation. So this glial activation is a measure of neuroinflammation, and we were able to dampen that down significantly with this mGlu5 NAM. Really interestingly, when we then dose this compound from later stages of disease, when we think the glial activation is driving disease progression, we can really improve quite significantly rotor rod function and extend the time taken for the mice to reach rotor rod failure. And this is probably one of the best molecules we've seen kind of late on in this disease model. So that's now in phase 1 trials in healthy volunteers, and we're obviously hoping that the data we've generated will kind of help push this towards ALS, motor neuron disease, as an indication. So just a simple PhD studentship, and we're continuing that collaboration now to work more on some of the mechanistic aspects. In terms of Keepstone, so this goes to the other end, so we've gone from PhD student all the way to a kind of early stage spin out, doing structure-based drug design around a novel target, which have very significant funding from Parkinson's UK, their virtual biotech program. So they are trying to stimulate all stages of drug discovery really, but fill that gap where, particularly when people are trying to take a novel approach in neurodegeneration, it's difficult to get the funding. So we collaborated with Parkinson's UK, set up Keepstone Therapeutics, and we're working on inhibitors of this NRF2 pathway, which is a transcription factor which activates the transcription of a range of genes that target multiple mechanisms that are relevant in neurodegeneration. I highlighted these early in motor neuron disease, but they're also very relevant in Parkinson's disease as well. And what we're doing is we're developing molecules that actually block NRF2 from binding to its ubiquitin ligase substrate adapter that would normally target it for destruction. So it's a protein-protein interaction. We're trying to make a CNS penetrant drug. It's not super easy or straightforward, but I think that's the interesting thing here, is we can take a slightly novel approach. And as I say, in both Parkinson's disease and motor neuron disease, the pathways that we target with this mechanism, and with some of the other molecules we're working on as well, we think cover a range of mechanisms that are really important in driving neuronal death. And we really like that approach in terms of being able to increase our chances of having a therapeutic effect in neurodegeneration. So I won't go through this in any detail now. I think I'm running out of time, but we've made some significant progress. There's still quite a bit of progress to make, and we're still seeking kind of partners and funding to take that forward. But one really key aspect of this, and this applies to all of our other programs as well, is that we've got a really nice translational biomarker strategy. So there's a whole host of things that we can do and methods that we're developing funded by other programs, where we can measure directly in the brain target engagement, or we can measure transcriptional target engagement biomarkers to really satisfy that translation piece. Okay, so that's all I wanted to talk about. Sorry, I think I may have taken a little bit longer, but this is my summary slide. I won't go into it in any detail, but I think the real difficult problem, I think, still in neurodegeneration is the sporadic disease that is the majority of ALS, of Parkinson's disease, of AD. And I don't think genetics are always going to give us the clue. I think we need really high quality pharmacology, good translation, more attempts on goal. I think that's my kind of key take home. So thank you, and any questions? These are the funders, obviously very indebted to all of our funders.